What if I told you that the most high-stakes copy job in the universe is happening inside you, right now? We're talking trillions of operations, all with zero room for error. So let's dive into this incredible biological mission, the perfect replication of your DNA.
So, just how high are the stakes? Well, every single time one of your cells divides, it has to perfectly copy several billion letters of its genetic code. That's like flawlessly retyping an entire encyclopedia collection in minutes. Yeah, one tiny slip-up, it could be a disaster. The mission, and it's not really one the cell can choose to accept, is simple on paper, but just staggering in reality. It has to create a perfect, identical copy of its entire DNA instruction manual. This is absolutely non-negotiable to make sure every new cell gets the complete blueprint for life.
The official name for this whole process is DNA replication. And really, it's the fundamental reason life, well, continues. From the simplest little bacterium all the way up to us, this process is how crucial information gets passed from one generation of cells to the very next.
Okay, so how in the world does it work? Well, it all goes back to Watson and Crick's original model of DNA. They gave us this beautifully simple idea called semi-conservative replication. Basically, the two strands of the parent DNA just unwind and each one acts as a perfect template to build a brand new partner strand. The result? You get two brand new DNA molecules, each one a perfect hybrid of old and new. Simple and elegant, right?
Well, that simple, elegant idea immediately slams into a wall of massive real-world problems. The cell faces a laundry list of challenges that make this whole copy job seem honestly pretty much impossible.
Let's just run through the disaster list, shall we? You've got the mind-boggling scale, you have to unwind a super tight helix without turning it into a tangled nightmare, you've got a main builder that can only travel in one direction, and, get this, that same builder can't even start the job on its own. And after all that, the final copy has to be basically perfect.
So how does the cell pull off this miracle? It does what any good mission commander would do. It assembles a molecular A-team. This is an elite squad of enzymes and proteins, and every single member has a very specific mission-critical job to do.
First up, you've got helicase. This is the unzipper. Its whole job is to just power down the helix, breaking apart the bonds holding the two strands together. It creates what we call a replication fork, basically prying open the DNA so the team can get in and read the templates. But as helicase unwinds the DNA, it actually creates another huge problem. The DNA ahead of it gets wound tighter and tighter, like an old-school telephone cord. That's where topoisomerase, the detangler, jumps in. It races ahead, makes these little strategic cuts to relieve the tension, and then seals it right back up. It's the master knot remover. Okay, next problem to solve. Our master builder can't just start building from scratch. It's a weird quirk, but it needs a little something to build onto. So the cell calls in primase, the ignition specialist. This enzyme lays down a short little strip of RNA called a primer, and that acts as the perfect starting dock for our main builder to get to work. And here it is, the star of the show, DNA polymerase, the master builder. This enzyme is a true marvel. It latches on, reads the template, and starts adding the right letters to build the new DNA strand at an incredible speed. It is ridiculously fast and accurate, but it has one major limitation. It can only build in one direction, what we call 5' to 3'. And that one little rule, that one-way only restriction, well, it creates the biggest, most complicated glitch in the entire operation. How do you copy both strands of DNA at the same time when they run in opposite directions, but your main machine only moves one way? I want you to picture a two-way street. The lanes go in opposite directions, right? That's exactly how DNA strands are. We call it anti-parallel. Now, imagine your road paving machine can only travel north. How in the world are you going to pave the southbound lane at the same time?
The cell's solution is both incredibly clever and, honestly, a little bit messy. For one strand, the leading strand, it's easy. DNA polymerase just cruises along behind the unzipper, building one long, continuous new piece. Simple. But for the other one, the lagging strand, it has to work backwards, building away from the fork in a bunch of small, separate chunks.
So on this tricky lagging strand, here's how it goes down. First, primase keeps jumping ahead to lay down new little primers as the DNA opens up. Then, DNA polymerase zips backwards and builds a short chunk, we call it an Okazaki fragment, until it bumps into the last primer. Then a cleanup crew comes in, removes the old RNA primer, polymerase fills in the gap with DNA, and finally, an enzyme called DNA ligase comes in and acts like molecular glue, stitching all those fragments together into one complete strand.
Sure.
Okay, so the copy is finally made, but the mission is not over. Not even close. Now we enter the final phase, quality control. We have to make sure this copy is perfect and that this whole process can keep going for generations without losing any vital information along the way.
Remember, accuracy is everything. And it turns out DNA polymerase isn't just a builder, it's also its own inspector. It has a built-in proofreading ability, a backspace key. If it accidentally puts in the wrong letter, it can feel the mistake, back up, snip out the wrong one, and put the right one in before it moves on. This little trick makes it 100 times more accurate. It's incredible.
But there is one last, sneaky problem. Think about that lagging strand, the one built in pieces. Once the very last RNA primer at the absolute tip of the chromosome gets removed, there's no way to fill in that tiny little gap. And this means that with every single cell division, our chromosomes get just a little bit shorter.
So how do we survive this? Well, we have these protective caps on the ends of our chromosomes called telomeres. In humans, it's just the same sequence, TTG, TGG, repeated thousands of times. This section is junk DNA. It doesn't code for anything important. It's a disposable buffer. So as the chromosome shortens, it's just eating away at this buffer zone, protecting all the important genes inside.
So how is this problem solved for the long haul, for our species? Well, in our most important cells, like our reproductive cells, the ones that get passed on, there's this amazing enzyme called telomerase. It's an end extender. It actually adds those telomere repeats back onto the ends of the chromosomes. It essentially resets the clock, making sure that every new generation starts fresh with full-length chromosomes.
And that leaves us with a really deep and fascinating final thought. This molecular process, this slow, steady shortening of our telomeres in most of our body's cells, it's basically a cellular clock for aging. So as we unravel these incredible biological missions, it really forces us to ask a bigger question. What does it truly mean to protect our code, not just for accuracy, but for our own longevity?